All right. So, continuing on the topic of error recognition. So just to kind of jump straight into it, assuming my laptop decides to behave. There we go. All right. So, this is a picture I made as a cocky little undergrad. You can see I'm very confident and happy about the image I made. I was very excited to get this first interferometric image. This was data that was taken way back in 2007. It's of M33 with a telescope called the Allen Telescope Array. I was super excited, super proud to get this image out. Spent a lot of time fiddling with things in Myriad to get everything just right, and you learned about color palettes and this, that, and the other, and just thought there's no way that, this was the best image possible. Clearly, I'm just the most modest person that you've ever met. And I thought this was really good. But as hopefully over the last couple of days you may have learned, this image doesn't exactly look maybe what you would expect. There are some things that look like they might be artifacts, things that look like you see these kind of negative bowls, other features that you're not sure whether or not they're physical or real. And so the leading question here that I'm trying to ask is, how can we make this image better? And can we recognize whether or not there are actually errors in this image that are maybe potential problems when they actually go to do science analysis with this data or this image that we've just created? So doing a very quick one-slide review of David's talk from yesterday. So at the end of the day, when you have your visibilities, you grid them, you do your magical Fourier transform, and you get this beautiful dirty image up top, and what we call sometimes the beam image, the PSF, the dirty beam. I've heard lots of different names for it, but basically this is the point spread function. This is what a point source will look like. You might notice that the beam image in this case is twice the size of the dirty image. That's not by accident. If you remember deconvolution or clean, if you're going around the map, you want to make sure that wherever inside the dirty image you are, you have the full size beam image to be able to subtract that to clean things off. So the beam image can and generally is larger than the dirty image that you generate. You run through deconvolution, and deconvolution spits out two things. Generally speaking, it spits out a clean model. So it hopefully does something in recognizing that, okay, there is some source of emission here with some priors, with some assumptions. In this case, I'm using clean. Clean assumes that everything can be broken down into point sources. And finally, out of deconvolution, you get some sort of residual map. Typically speaking, when people talk about what are called clean maps, or the final end data product of the interferometer, you're looking at the summation of the clean model plus the residuals. So almost every single interferometric map that you've ever seen is the one that's on the far right-hand side. So if you look at these things that are up here, you might natively assume that the thing that's the most important thing for all of your analysis is the image that's on the far right-hand side. And I'm here to tell you, no, it's not actually the case. The thing that's the most important map out of all of these is the residual map. The reason why is because the whole point of doing clean, of doing deconvolution, is to basically generate some sort of model of what the sky looks like. And so the clean model is the thing that you think the sky actually looks like. The residuals are basically telling you how good a job you did pictorially. And so you know that if you see things that are weird, if you see things that don't look like random noise, your deconvolution, your modeling has gone awry at some point. And so the question is, can we recognize where something went wrong and then fix that? So one point to make here briefly, if you look at this image, if you look at this residuals map right now, you might actually see something that looks like structure, right? Like I see this kind of regular patterning of these dark spots. There's some regular patterning of these bright spots, these red spots. And you might look at this and say, oh, Karto, you've already got a problem. This is a perfect demonstration of something going wrong. This is kind of a little weird and deceptive. The problem is that, of course, if we had a perfectly random map, then yes, you would expect something that just looks like TV static, where points are completely disconnected and it just looks completely random. Problem, of course, with an interferometer is that you have the interferometer acts as a spatial filter on your image. So there are modes in this image, modes in the noise that are missing. So the closest point of comparison that I can make, it's like listening to white noise versus brown noise. And I thought, well, maybe I'll play something off of Wikipedia, but then I realized I can actually do this with this audience. So white noise is like, red noise is, right? You hear that there's a difference between those two things, and if you're comparing them side by side, you might think, oh, that thing has the pattern in it, that thing is missing something. And you're right, it is missing something, but it's not, it's missing something because it's missing some set of frequencies, not because there's some artifact or some weird thing that's there. So your eye can be a little deceptive when you're looking at these images. So everything I'm about to tell you right now, just take a little bit of warning. You're looking for things that are easy to see with your eye, but you're always looking to back things up, to move back to the visibility domain, to look at gains, to look at whatever, to check and see where the actual source of the error is. All right, so here's my lovely, lovely point source at Field Center. I've spent a lot of time imaging this guy. At the end result, so this is a 1 Jansky source. I've clipped it down to make some of the artifacts a little bit easier to see. And I see that there are these weird errors, these weird negative bowls on either side of the image. So that's the clean plus the, that's the clean model plus the residuals. Let me just look at the residuals for a second. And yeah, actually, if you look very carefully, you can see very clearly that there are symmetric errors around the center, around this point source. Usually, when you're seeing symmetric errors, you are generally pointing to some sort of problem in the amplitudes. So there's some sort of problem with the amplitudes. In this case, it's an antenna amplitude error. So some data for some antenna got scaled up too high. The way that this affects the image is that the data belonging to the points on this red track are too high kind of on these sides, and so you end up with this symmetric artifact, right? Because the data is, if you've got something that's, what is it, symmetric in that domain, it's going to be symmetric in that domain, or a real only in that domain, it'll be symmetric in that domain. And so you'll see these positive artifacts that look something similar like the beam, and in fact, the artifact will basically have the beam shape, if you will, of you only included those baselines to that one antenna that had this particular corruption. So, different error now. I'm looking at something that looks anti-symmetric, right? Bright spot here, faint spot here, bright spot, faint spot, bright spot, faint spot. Anybody want to guess what this might be caused by? Phase, there you go. Phase, that's right. Things that are anti-symmetric generally point to things that are complex conjugates of one another, but are not actually duplicates of one another in the UV domain. So again, this is a single antenna, antenna 1, that happens to have a phase error. Phase errors will produce symmetric errors in the imaging domain. And so if you see things that look like symmetric errors, they can be pointing towards phase errors, either in the baselines or, generally more speaking, phase errors in the antenna gain solutions themselves. So if you see symmetric errors, check out your amplitudes, particularly for your gain solutions. If you see anti-symmetric errors, check your phase solutions. This, I feel like 90% of the time, will catch basically most of the errors that you're looking for. You'll see these residuals around sources in your field of view. This is easier to do with point sources, obviously, than very extended structure, but you'll still see symmetric or anti-symmetric features. All right. So, time for a quiz, because that's really the thing that you need after however many hours of lecture. So, I've thrown up an error here. This is the residuals map after doing basic cleaning. It's the same point source. Anybody want to guess what kind of error this is?
Amplitude.
Amplitude. Very good. Why amplitude?
Everything seems symmetric.
Everything seems symmetric. And in fact, everything looks kind of, it's a little bit weird It's especially weird if you do one thing, which is if you actually look at the point spread function, it looks very similar to what the point spread function looks like. In this case, the problem that we're seeing right now is the fact that it's actually two things combining into one. The first of which is basically that this is looking at a quasar. We kind of hand-wavingly assumed that quasars have roughly the same flux over the frequency band of interest. But as you go over wider and wider bandwidths, and the SMA is actually starting to get to the point where the wide enough bandwidths are actually starting to be a little bit of a problem with this, you have different amounts of flux at different frequency channels. And so the key thing here is that when you made your image, when you combined everything, you made this general assumption that all of the data that went into that image is sampling the same thing in the sky, right? Now, you don't have to do that for image. There are ways of getting around this, different techniques. But just know that that assumption can be wrong, and it can be wrong on two accounts. One is particular frequency differences. So just because it doesn't even matter if it's a very relatively narrow range of channels, right? Because there's spectral line emission, there are things that are very, very narrow. You can get very weird and strange imaging artifacts when you have a patch of sky that has a different response at a bunch of different frequencies that have been all averaged together. Generally speaking, if the sky is changing in some way, you should make a different image for it. The other thing, as Mark and David actually pointed out yesterday, I think, you might assume that sources in the sky are static with time, and they most definitely aren't. And it's not just quasars. There's masers, there's all sorts of things that go boom in the night, right? And so it's not a guarantee that you can combine data sets from different days if your source is rising and falling in terms of its emission strength. So whenever you're imaging, take a lot of care with both of these cases where either you have frequency structure or time structure in what it is that you're seeing in the sky. So this is just from the source side. There's also two limitations that come in on the interferometer side itself. And that's the problem, if you want to call it, that we can't record an infinite amount of data. Is Mac still in the room? All right, perfect. We can record an infinite amount of data. No, Mac is our systems administrator, who I think regularly sees how much data I record on a semi-regular basis. And I'm always thrilled, I know. But no matter, at the end of the day, you're always averaging over something, right? You're always averaging over a frequency range, and you're always averaging over a time range. Now, it can be hertz that you're averaging and seconds that you're averaging over. But the key thing is that in the frequency domain, the resolution of the interferometer changes as a function of frequency, right? So if you average together multiple different frequencies, you'll get this thing that we call bandwidth smearing, which is that the interferometer, this is the thing I talked about the very first day, I talked about interferometry. You'll have different things kind of sort of interfering with one another because they are sampling slightly different spatial modes in the sky. And the bigger and more extended your interferometer is, the more of a problem that this becomes, right? Because that wavelength scaling depends on baseline distance. So if you have super long baselines, this can actually be a big problem for your interferometer. The other problem is a little bit more subtle, but that's the time averaging piece. So you kind of see this repeating pattern. One time interval in this case is one repeating pattern. So you see it goes kind of red, blue, green, red, blue, green. One time integration is kind of red to red. And so you can see that each integration is kind of stretching over a range in the UV plane. It's not just an infinitesimally small point. And that can also introduce some interesting, weird artifacts that depend basically on how quickly your baseline positionings are changing as a function of time. So to give you an idea of what these things actually look like, and this looks sort of okay on this image. Let's see if I can turn this down. It's pointing at a lot of pictures. It's exactly the wrong one. Yeah, all right, it's fine. I tried. That's the important thing. All right, so time smearing. So here you can see, so this is a good cleaned image. It looks really nice. It looks really good. You just see the point sources. You've got really good eyesight. You might see a little speckle of something, but everything looks really good. Time smearing, things have kind of gone wonky and all over the place. And in fact, it's a bit, these things that should be point sources are extended in different ways and in very different, have now new and interesting geometries. The time smearing is a little bit difficult to predict because the rate at which an individual baseline is moving through the UV plane depends a lot on the 3D geometry of the array. So you're looking at how quickly a projected 2D geometry is changing because of the 3D geometry. So time smearing artifacts can be problematic to recognize. It just kind of looks like everything's crappy and blobby. Bandwidth smearing is at least a little bit easier to recognize because with bandwidth smearing, everything will kind of point towards the center. So if you have point sources that look like they're all pointing towards the center, usually means that you have a bandwidth smearing problem. You can fix this by using a technique called multi-frequency synthesis. I think there's an MFS switch in CASA. There's also one in Myriad. There are modern tools for taking care of this particular problem, but it is something that you have to enable, and it does require a bit more processing time. All right, so anybody want to guess what this is? Think back to your Fourier transforms. So the point source there is good, right? But there's this stripy pattern that's going through the field of view. What might be wrong?
Your delays.
Say again?
Maybe your delays are off.
Delays are okay because I've still got a point center, and I would expect something that looked closer to like an antenna based, like a symmetric error, and this is not. And it looks kind of sort of symmetric. Any other guesses? David.
One high amplitude point.
One bad data point. One bad data point, right? A single impulse in the visibility domain produces a rippling pattern in the image domain. So I've highlighted this one little red point that's up there. Because the sky is real, each interferometer ends up containing two different pieces of information. So a bad piece of information pops up in two different positions in the UV plane. But this all really comes from one bad measurement. So one bad measurement will produce this rippling pattern. So if you see this, it means you got to go hunting for the one bad point. This is the kind of thing that's usually pretty easy to find. All right, let's go tougher.
Ooh.
Oh, yeah. Any guesses?
Too bad point.
That is a good guess. But I would expect two bad data points to look like a ripple going this way and a ripple looking like that way, right? There is clearly some rippling structure that's here, but it's kind of curving around, and maybe a hint, looks like it's maybe the summation of multiple nearby bad points, right? Any other guesses? No guesses. Oh man. What's one thing that can, or what's one interval of data, I guess?
Baseline.
Bad baseline. There you go. So a bad baseline can produce this kind of weird, what you're kind of sort of seeing is a kind of slow beating pattern of multiple fringes, one against another. And so usually if you see this kind of pattern where it's a ripple, but it's kind of a weird complex ripple and there's some curving structure thing, it means that you've probably got a bad baseline. All right, next. Here's this blobby thing. There's kind of a sort of symmetric-ish type of error, and it looks a little bit like the point spread function. Anybody want to take a guess? What's another interval of data or block of data?
Bad antenna?
It's a good guess. No. In fact, it almost looks like, and this may be a hint, because it looks like the PSF, it almost looks like all the antennas are bad.
A particular time interval.
A particular time interval. Yeah, so this is kind of The reason why they look very similar is because a bad integration is kind of a small subset, small sampling of the entire UV plane. So if you have something that looks similar-ish but not quite to the PSF, sometimes it's pointing towards just a bad single integration. All right. So, moving on. That's the end of the quiz part, I think. We've left the land of bad data points, and we're now going on to deconvolution. So you get this image, it pops out. You see there's kind of lots of regular repeating structure, and you're not quite exactly sure what's going on. And I don't think anybody talked about this yet, so this is kind of cool. So you check your residual image, and wouldn't you know, it looks like you've got emission at each one of these positions. But it doesn't look like there's any weird errors or anything. It actually just looks like there's still point sources that are there. So this is referred to as undercleaning. So basically your model has underestimated the amount of flux that's there. Kind of more modern instantiations of clean will look at the map and hopefully won't stop until things fall below some threshold. But one reason to check your residual maps is to make sure that the deconvolution actually cleaned out all of the flux that's inside your map. And so if you see something that looks like almost a carbon copy of your what your clean map looks like, it may actually be that deconvolution didn't run all the way to completion, or you didn't provide it enough iterations to be able to converge on a good solution. All right, now on the complete opposite side. Oh man, this doesn't show up. That's a bummer. Actually, maybe some of you can see it, but there's these little speckles that are up on the screen that weren't there before, that weren't there in the perfectly imaged domain, and they're all over. And in fact, if you were to look at this image when you go home, or when you look at these slides online, you'll see that this looks like an amazing image, but when you look at the residual map, I think it's a lot easier to see. What in the hell is going on there? So there was a question that was asked during the break, is there anything, can you ever clean too much? The answer is yes. And it's not just because I'm a slob. You can actually basically clean out noise or be creating artifacts, because clean is effectively locking onto noise in the image and is trying to deconvolve. And you end up with this weird kind of corrugated pattern in the residual images that will pop up also in your clean images. So if you see really lots of like very high frequency structure, lots of detail, lots of things that look like individual point sources on a very weird background, and you see something like this in your residual map, sometimes it's a sign that cleaning has gone on for too far. And this is kind of if you're lucky. If you're not lucky, clean will basically just explode. And because it's a nonlinear process, it is capable for it to go off into infinity and basically just leave you with something that has a lot of nans and zeros and weird things.
This is like, so you didn't draw any mask here. I mean, just
correct.
To tie in with what I showed earlier.
Yes, I did not draw a mask here.
Like this, if you do it blindly and you're basically setting 1,000 iterations and you leave it free to go pick peaks.
That's right.
So that's part of the reason why you choose masks. So
right.
Go off and do this.
All right. So last thing. So this looks kind of cool, right? This kind of windswept something flaring off into the distance. But clean, obviously, well, when clean went through this, it detected something that was point source here and something that was point source here. And it's all this other structure, and I think it cleaned some of it out. But you have something that looks really regular, it looks really patterned. But it's just, yeah, this doesn't look physical, right? It looks like a chain link fence, basically. So the key here is that there's a point source right here. And if you notice the axes here, the point source is right about here, basically just outside the field of view of this image. So in this image, I only imaged out to 1 times the primary beam width or the field of view. In this case, I imaged 2 times out. This is part of why you always image 2 times out, because if you don't image it, you can't clean it. And if there are point sources that are casting emission into the thing that you're interested in the center of field of view, those the emission, the point spread function from that source won't really be properly cleaned out, and you'll be left with a map that looks beautiful but is worthless for science. So always image a larger field of view than what you're interested in, because there may be things around the periphery that will give you a bad image.
Karto.
Yes?
But it is time expensive to make a very large image.
It is, yes. So you notice that in this case, I generally go by the rule of 2 times the full width half max. That gets you down to about 5% of the primary beam max. I think CASA default is 10%. Myriad doesn't tell you anything. It lets
Now, CASA, you choose how big you want it.
Okay, all right. So yeah, I think 2 times is a safe amount, at least to do once. You don't have to do it for all the frequency channels, but at least for your continuum image, make sure that there's not some thing lurking off in the distance that you're having. And then the final point that I just want to make here, when you're making an image, of course, you are making a model of the sky. We talked a bit about, you don't know what your source looks like, and so sometimes it's difficult to look at the visibilities and know what to do. But if you've made a model, you can subtract off that model or otherwise do other sorts of interesting or statistics with that data, and actually go hunting for problems, even if your field is relatively complex. So if you subtract off some data and you notice, hey, this one baseline, okay, it looks kind of noisy compared to all these other baselines, but when I fit for a model and I see this big thing pop up, that may actually be a good sign that the other baselines are telling you that, okay, something is weird about this particular data point, this particular data set. Obviously, whenever you do this, use a lot of care. You can inadvertently force your image to be more and more correct, or to converge on a solution, I should say, that isn't actually correct. But this is a nice way to get around the problem of not knowing what the sky looks like when you look at the visibilities, because this has the potential of removing the source emission so that all you're looking at is blank noise, and blank noise is, or should be at least, very statistically easy to describe and to look for outliers. So that's all I've got, so I'll stop there and I guess take any last questions that might be lurking. Yes.
Just to clarify, that full width half max is of the expected beam size?
That is the primary beam size, not the synthesized beam size. So that's so for the SMA at 230, it's about 50-ish arc seconds across. At 345, it's about 30 arc seconds. So typically I would image over 100 arc second field of view at 230, just to be sure that there's not something weird just adjacent.